Exploratory behavior in an empty arena seems to be stochastic. Nevertheless, it does not mean that the animal is a random walker. The physicality of the world in which the animal operates, its anatomical structure, physiology, perception, emotional states, and cognitive capabilities all determine the statistical boundaries of the behavioral space within which the animal can operate. This is a small part of a real trajectory by a mouse while exploring an empty arena. Imagine that we take all positions visited by this mouse, but relieve all constraints on how we connect these coordinates. Not surprisingly, connecting positions randomly yields haphazard behavior that does not show any resemblance to mouse locomotion. Now let's start adding some reasonable constraints. For an example, a mouse lives in a physical world where step size and speed are limited by its anatomy and its muscle strength. Incorporating such limitations on the possible connectivity of visited positions changes the motion characteristics of the possible outcome trajectories one can get. Inertia limits the mouse's ability to change direction in high speeds, further reducing the motion range of the motion variables. Finally, emotional constraints may prevent the mouse from covering evenly the large arena, as can be seen once returning to the real mouse data, blue dots, showing a strong preference to the walls and the home base. By finding more constraints, one can regain the behavioral qualities that define the mousiness of this animal. Inspection of the mouse locomotion reveals frequent occurrences of what seems like high mirror symmetry. A measure of mirror symmetry was defined as the arc length of trajectory over the minimal area between the two successive parts of this trajectory rotated one against the other. This allowed a more thorough inspection, scanning several kilometers of mouse trajectory in various arc lengths. To avoid trivial cases of symmetry, movement along the wall was secluded, and only arc lengths that are much longer than the 8 centimeters mouse length were tested. For each range, the peak mirror symmetry was selected. This peak defined the corresponding position along the trajectory and arc length to be considered. By using this method, many more examples of high mirror symmetry in relatively long motion segments were found. In order to test whether these findings are just some peculiar coincidence, a similar extensive scan was made, only this time the two motion segments were not adjacent in time. Symmetry values were found to be significantly lower in this latter condition. This suggests that these mice tend to frequently mirror their motion segments in what seems like using a short duration memory storage of their motor actions. To summarize, behavior can be considered as the subspace that remains after secluding all actions that are not available to the animal due to constraints. The physicality of the world in which the animal operates, its anatomical structure, physiology, perception, emotional states, and cognitive capabilities all determine the statistical boundaries of the behavioral space within which the animal can operate. The very signature of being a certain creature is reflected in these limitations that shape its behavior. A major goal of ethology is to expose those constraints that carve the intricate structure of animal behavior and reveal both uniqueness and commonalities between animals within and across taxa. Here, yet another such constraint was added to the ethological knowledge.